Learning outcomes. After studying this module, you will be able to know about the significance and principle of spectroscopy, various spectroscopic techniques and their methodology, and the importance of spectroscopic technique in detection of poisons. So, dear students, let us start with the introduction of this module. Spectrophotometry is the quantitative study of electromagnetic spectra. It could be defined as the branch of science which deals with the study of interaction of electromagnetic radiation with matter. Spectroscopy is one of the most prevailing techniques available for the study of comprehensive range of samples. Spectrophotometry is concerned with visible light, that is near ultraviolet and near infrared. It consists of the use of a spectrophotometer. A spectrophotometer is a photometer, that is a device for measuring the light intensity, and that can measure the intensity as a function of the color, or more specifically the wavelength of light. There are several categories of spectrophotometers. Among the most important distinctions used to classify them are the wavelengths they work with, the measurement techniques they use, how they acquire a spectrum, and the causes of concentration. Other significant features of spectrophotometers comprise the spectral bandwidth and the linear range. Possibly the most general use of spectrophotometers is the calculation of quantity of light absorption or the light absorbed. There are two main classes of spectrophotometers, that is single beam and double beam. A double beam spectrophotometer deals with the ratio of the light concentration on two diverse light paths, whereas the single beam spectrophotometer deals with the absolute light intensity. Although ratio measurements are easier and generally stable, single beam instruments have advantages. For instance, they can have a larger dynamic range. The spectroscopic approaches are distinguished as both atomic and molecular, based on whether they apply to atoms or molecules. Along with that distinction, they may be categorized on the nature of their interaction. Absorption spectroscopy takes the range of the electromagnetic spectra in which a material absorbs. This comprises of the atomic absorption spectroscopy and numerous molecular methods, including infrared spectroscopy in the infrared region and nuclear magnetic resonance spectroscopy or NMR spectroscopy in the radio frequency region. Emission spectroscopy applies the array of electromagnetic spectra or EMR spectra where a material discharges or emits. The material first essentially takes up the energy. This energy may come from a range of sources. Molecular luminescence methods comprise of spectrofluorimetry. Scattering spectroscopy deals with the quantity of light which an element scatters at definite wavelengths, incident angles, and the polarization angles. The scattering procedure is very quicker than the absorption or emission process. One of the most valuable uses of light scattering spectroscopy is Raman Spectroscopy. Next, we will be studying about the significance of spectroscopic technique. Spectroscopy is the most influential instrument accessible for the study of atomic and molecular structures and is applied in the examination of widespread samples. Analytical absorption spectroscopy in the ultraviolet or UV and visible regions of the electromagnetic spectrum has been extensively used in pharmaceutical and biomedical examination for measurable purposes and with some restrictions for the classification of drugs, impurities, metabolites and associated elements. By contrast, luminescence methods and fluorescence spectroscopy in particular have been less widely exploited despite the undoubted advantages of greater specificity and sensitivity commonly observed for fluorescent species. However, the wider availability of spectrofluorimeters which are able to show modified excitation and emission spectra coupled with the detail that dependable fluorogenic reactions permit non-fluorescent species to be examined fluorimetrically has led to a renaissance of interest in fluorimetric methods in biomedical analysis. Next, we will move on to the types of spectroscopic techniques. Here, first we have the UV visible spectrophotometry. Absorption spectrometry in the UV visible region is considered to be one of the oldest physical methods used for measurable examination and structural elucidation. The laws of absorption of radiant energy with which the analyst is concerned involves the relationship between the magnitude of absorption and the quantity of absorbing material present in solution. These laws are applicable to absorption energy in any region of the EMR spectrum or the electromagnetic spectrum. The absorption by matter of electromagnetic radiation in the domain ranging from the near ultraviolet to the very near infrared between 180 to 1100 nanometers has been studied extensively. This portion of the electromagnetic spectrum designated as the UV or visible, since it includes radiation perceptible to the human eye, generally yields little structural information but is very useful for quantitative measurements. The concentration of an analyte in solution can be determined by measuring the absorbance at some wavelength and applying the Lambert-Beer law. The region of the spectrum is conventionally divided into three subdomains termed as UV, that is from 185 to 400 nanometers, visible, that is from 400 to 700 nanometers, and very near infrared, that is from 700 to 1100 nanometers. Most commercial spectrophotometers covers the spectral range of 185 to 900 nanometers. The origin of absorption in this domain is the interaction of photons from a source with ions or molecules of the sample. When a particle absorbs a photon from the UV visible region, the analogous energy is taken by one of its farthest electrons. Next technique we have is the atomic spectroscopy or the absorption and emission. There are two types of atomic spectroscopy. One is atomic absorption and the other is atomic emission. The absorption of energy by ground state atoms in the gaseous state forms the basis of atomic absorption spectroscopy. When a solution containing metallic species is introduced into a flame, the vapors of metallic species are produced in the atomic state. Some of the metal atoms may be raised to an energy level sufficiently high to emit the characteristic radiation of the metal. But a large percentage of the metal atoms will remain in the non-emitting ground state. These ground state atoms of a particular element are receptive of the light radiation of their own specific resonance wavelength. Thus, when a light of this specific wavelength is applied, part of that light will be absorbed by the atom of the element and the absorption will be proportional to the population of the atoms in the flame. Thus, in atomic absorption spectroscopy, one determines the amount of light absorbed. In atomic emission spectroscopy, which is the second type of atomic spectroscopy. A sample is excited by absorbing thermal or electrical energy, and the radiation emitted by the excited sample is studied for qualitative and quantitative analysis. Emission spectroscopy is regarded as the most reliable method for elemental quantitation. Analysis by atomic emission spectroscopy constitutes a general method for measuring elements on the study of the radiation emitted by their atoms present in an excited state, usually following ionization. Several procedures are used to break down the samples into their constitutive elements, and most notably by the effect of high temperatures, plasmas, spark sources or glow discharges. The emission being far more complex than those of flame emission, instruments require very high quality optics to resolve interferences from both spectral lines and matrix effects. The third type is infrared spectroscopy or IR spectroscopy. Analytical infrared studies are based on the absorption or reflection of electromagnetic spectrum or radiation that lies between 1 to 1000 meters. This is one of the most common spectroscopic techniques used for compound identification and measuring of concentrations in many samples. This spectral range is subdivided into three smaller areas: the near infrared, the mid-infrared and the far infrared. Although the near infrared is poor in specific absorptions, it is considered as an important method by quality control laboratories for quantitative applications. By contrast, the mid-infrared region provides more information upon the structures of compounds, and consequently it is much used as a procedure for identifying organic compounds, for which it remains a form of functional group fingerprinting. The far infrared or far IR region requires the use of specialized optical materials and sources. To conduct these analyses, there is a full range of instruments from Fourier transform spectrometers to a multiplicity of analyzers of dispersive or non-dispersive types, specialized in the measurement of predefined compounds, example analysis of gases and vapors, or which allow continuous analysis on production lines. The Fourier transform infrared spectrometry offers numerous possibilities for the treatment of spectra and has applications for the analysis of structured microsamples, including the infrared microanalysis. When a molecule is subjected to infrared radiation, transitions take place between rotational and vibrational energy levels in the ground electronic state. These transitions give rise to an absorption spectrum characteristic of the compound. This is in contrast to ultraviolet radiation, which also causes transitions between rotational and vibrational energy levels. But because of its greater energy, these transitions occur at higher energy states. In infrared, the absorption occurs only where a chance of dipole moment of a molecule can take place. The fourth type is Raman spectroscopy. Raman spectroscopy involves the interaction of light with matter. It is a non-destructive technique based on inelastic light scattering, that is, the energy or the frequency of the incident laser light is changed or shifted. The difference between the incident and scattered frequencies corresponds to an excitation of the molecular system, most often an excitation of a vibrational mode. By measuring the intensity of the scattered light as a function of the frequency difference, we obtain a Raman spectrum, which reveals information about a sample's chemical structure and the physical state. Raman spectrum is similar to an infrared spectrum, though Different vibrational modes are observed in these two complementary techniques. Raman spectroscopy provides quick, easy and most importantly non-contact and non-destructive analysis, both qualitative as well as quantitative, with no sample preparation required. Samples may be tested directly with fiber probes or through glass, quartz, sapphire cells and fibers. Forensic applications of Raman spectroscopy includes identification and analysis of explosives, inflammables, toxic solvents, additive drugs, drugs of abuse, biowarfare agents and angstrom documents. The fifth type that we will now study is mass spectroscopy, or better known as MS. Mass spectroscopy or MS is an analytical method of characterizing matter based on the determination of atomic and molecular masses of individual species present in the sample. The instruments employed for carrying out mass spectrometry can be classified into different categories according to the mass separation technique used. A minute quantity of sample transformed in the gas phase or in a suitable form is ionized, and the resulting charged species are then submitted in an enclosed space maintained at high vacuum to the action of an electric and or the magnetic field depending on the type of instrument. The study of the forces exerted upon the ions allows the determination of their mass to charge ratio, that is M/E ratio, and eventually their nature also. This method destroys the compound sample, but owing to the great sensitivity of the technique, only a tiny quantity is required. The results are displayed as a graph called mass spectrum and presented in the form of abundances of ions formed and classed in increasing order of their mass to charge ratio, or M/E ratio. Continuous improvements have been made, and mass spectroscopy has been a most versatile, sensitive and widely used analytical method that is available today. Miniaturization and the emergence of new ionization techniques allow this method to be present in a variety of sectors: organic and inorganic chemistry, biochemistry, clinical and environmental chemistry and geochemistry. This technique is found in many industrial control processes as well as for regulation compliance. The sixth technique that we will study is Xray Fluorescence spectrometry, or XRF. X-ray fluorescence is an atomic spectral property universally recognized as a very precise technique currently employed under this name to provide qualitative and quantitative information on the elemental composition of a type of sample or all types of samples. The principle of operation involves irradiating the sample either with an X-ray beam or by bombardment with particles such as electrons having sufficient energy in order to observe the resulting X-ray fluorescence, or XRF, emitted by the sample. The universal character of this phenomenon, as well as the possibility to rapidly examine samples most often without preparation, explains largely the success of this non-destructive analytical method. The energy dispersive X-ray fluorescence technique finds application in the analysis of metals in soils and sediments and environmental samples of air and water. However, attempt to extend the methods to environmental samples under different projects met success only with soil samples. The energy dispersive XRF technique is rapid, non-destructive, requiring only sample preparation by microwave digestion or pulverization of sample. As it does not require drastic reagent or additional waste stream, this is a safe technique. The entire method is electronic data based and very fast. mercury, arsenic and selenium are considered volatile elements and some care must be taken in their analysis, especially in case of mercury. ETXRF in conjunction with the fundamental parameters software is an excellent method for rapid screening of mercury contaminated soil. Now students, let us summarize this module. Spectrophotometer is the quantitative study of electromagnetic spectra. Spectrophotometry deals with visible light, near ultraviolet and near infrared. Spectroscopic methods are much more rapid and much less time consuming. They give information which is recorded in the form of a permanent chart, generally in an automatic to semi-automatic manner. The structural information gained by spectroscopic methods or techniques is much more precise and reliable, as they are highly reliable in establishing the identity of two different compounds. They are much more selective and sensitive and are extremely valuable in the analysis of highly complex mixture and in the detection of even trace amount of impurities. Atomic spectroscopy is concerned with interaction of electromagnetic radiation with atoms which are commonly in their lowest energy state called the ground state. Molecular spectroscopy is the communication of electromagnetic radiation with the molecules. This results in transition between rotational and vibrational energy levels in totaling to electronic transitions. Thank you.